divided up into like the NHST side part that's up in Ninewells, and more the basic research part that's based here. And so I'm in basic research, and my group is, for example, interested in taking a look down the microscope, looking at the cell and seeing what happens if we disrupt like a small part of the cell, and like what kind of consequences that has for the whole cellular machinery. And Noir is more into very, like, more zoomed in into like what happens when you change like the one single
MRC protein phosphorylation unit upstairs, if I may interject.
Go on,
Yes.
go on.
Yes, well, my lab focuses on a class of proteins called kinases. Kinases have a sole function is to transfer phosphate group onto other proteins, thereby conferring different functions to that protein. So this phosphorylation step is sometimes involved in cascades, sometimes called signaling cascades. And it has long been established that for a cell to start dividing uncontrollably, it's down to aberrant phosphorylation. For it to evade the normal constraints of cell division to proliferate, it has to be down to aberrant phosphorylation. That's why kinases are very attractive targets for drug companies. So that's why what my unit focuses on. But I personally don't work on cancer. I work on a kinase that's involved in a neurological condition that causes a movement disorder. But the bottom line is, I'm interested in kinases. So yes, there are about 500, more than 500 kinases in the human genome. So that's why drug companies are really interested in them. So every signaling pathway in a cell is bound to be affected by a kinase. So that's what we'll attempt to explain to you. So if you have any questions, please ask. Would you like to carry on?
Yeah, so we're going to take you on a tour through the labs. None of us are working in the actual labs we're taking you through, but our labs are basically organized pretty much the same. So we're taking you through the ground floor, and that's mainly the drug discovery unit, which is obviously also important. Like, they work more on parasites and disease, but obviously drug discovery is also very important for cancer treatment. And yeah, well, like, going through the labs, we're going to explain like some of the equipment to you that we're using on a daily basis. And on the tour, you're going to have to wear lab coats and safety specs, because we're also taking you through some of the organic chemistry labs, and there it's like important to have safety specs on. And then may I ask you to maybe not like touch too many of the things, because like, especially for example, like the pipetting robots, they'd be sensitive to like any kind of impact that is like on the tables or something. So yeah, just don't touch everything, basically. Yeah, but yeah, should we just?
Yeah, we should go. Just to point out, all the doors are protected by swipe cards. So if you lag behind, you may be stuck behind the door. So try and stick together as we walk through the labs. So that's about it. Yeah.
And if you want to leave your
I just tell my mom that, and she like one of those actually get exactly what the scientists
Get them to maybe TV first. And, yeah. Yes.
pathogens that require special containment. They're called cat-free suites, and they're monitored all the time in case something happens to anyone in there, so that people can get like help. It's just a safety.
Not the doors in case.
Any particular aspects of the cancer and biochemistry you're interested in?
So these are like so if you work with bacteria or also just cells and you want to avoid contamination, then you work like under a bench like that has like a special airflow so that no air from outside would get in and possibly contaminate your sample, or nothing from inside will come out to cause a hazard to you. But that's only important, of course, if you work with very contagious things, which I don't, but these guys here do.
Do they rate labs here based on how hazardous the material you're using?
Yeah, you have categories, so...
Because I've done the biosafety course before and
right.
do that here as well.
Yeah, so they have this one cat-3 suite where they work with trypanosomes. That's like the parasite that causes sleeping sickness. So, yeah, that's the most pathogenic organism we work with. But most of the scientists will work with human cells or even like Drosophila, fruit fly, like that. But actually, I think
This one will cost about 50 grand. That's a rudimentary kind of model. The bigger ones are about $1 million.
Whoa!
Yes. They have different properties. Okay. Different properties depending on what you want to do, what kind of proteins you want to study. On that basis, you decide which kind of machine to use. There's different variants of it. So, for example, I work on brain proteins. I'm studying neurological condition. Okay. What's the next stop?
we go left,
Yes.
and anything that so this is another specialized room. This is the room where people work with radioactive material, and that's very important to be able to label, for example, proteins. And the levels of radioactivity people deal with is obviously not high, but still.
It's a health hazard, so
yeah.
it's very tightly regulated. That's why some labs like this one have a specialized room dedicated to it, so that the radioactivity is contained in a way.
Yeah, but the benefit of it is that obviously if you have, for example, you work in protein phosphorylation, if you have your phosphor residue labeled radioactively, and you want to find out if your protein of interest gets labeled with this phosphor residue, you can just look for the radioactivity, and that's much easier than trying to find the phosphor residue on the protein. And that's like a really nice amplification step. That's why it's used very extensively in biochemical research. Yeah, just watch it for a while and be impressed, I guess.
Will you stay here for hours?
So they're kind of programmed to pick up their pests and then in those microliter formats kind of have different volumes of, for example, a drug, and like put it in, and then you can see like by looking at the outcome, like the effect on the cells, what different volumes, what different amounts of drug will do to a cell. and then thereby determine, for example, the effective dose of the drug. Yeah, but I have never worked with this. I assume it's very complicated. It does. There should be some sheets around here to like indicate how much every piece of equipment is. I've seen them lying around. Maybe it's like we're going up the other one, but it's actually.
Okay, draw your attention to this high-tech piece of equipment here, called the operating microscope. Here what the researchers do, they screen they're studying this infection. Okay, what's colored in red on this picture are human macrophages, which are immune cells. Okay, and what's in purple is the nucleus of the macrophages, and the green dot is the parasite they're studying. Okay, what they're trying to find is a compound that will, you know, kill the parasites, but not the macrophages. So they stick them in this 384 well plates, so they can do like 384 tests at once. So this machine can count, it can be left over the weekend, and it can work by itself basically, and it's really efficient for these guys who work down here.
Nice shape. Have you seen the cell made of balloons? And the pink part is the nucleus.
They get a big grant and that's back. So now, what we have to work out is whether it four young scientists come hope to get so many things published, they expect to get so many things published per grant. So much to the public.
Now they've got the slime on. So that's why bacteria like making this slime. They like making biofilms because it protects them. I can mean to help, but I can try and help. But actually, the best way of getting rid of a biofilm, just imagine you had a biofilm on your teeth. It's just to brush it off.
And then get all the glucose. Brush it off with your toothbrush.
So you know you have to brush your teeth twice a day. This is why. Because if you don't brush all those folks' bones,